here. Thanks everybody for joining us today. I just want to remind you that this seminar is being recorded today. And so please let us know if that's an issue. For those online, feel free to turn off your cameras if you would like to. I'll start today by an acknowledgement of country. As we do, I'd like to, of course, acknowledge the traditional owners of the land here in Canberra, the Ngunnawal and Ngambri people. I'd like to also extend that acknowledgement to the traditional owners of the land, to anybody who is joining us from online, and pay my respects to their elders past, present, and emerging. We also, of course, want to acknowledge that the sovereignty of this land has never ceded, and it always was and always will be Aboriginal land. Today, we're very excited to have two great presenters. We're going to begin with Sumeet Rai, but before I do that, I also wanted to just extend a quick congratulations to Jessica Donaldson. Jessica is a PhD student, and she recently published a paper titled "Multiple Group Memberships Protect Against Anticipatory Anxiety for Social Situations." So very big congratulations to Jessica, and please a reminder, if you have students or other staff that you know of that we want to extend a congratulatory message out to, we're absolutely excited and thrilled to do that during our seminar series. So today we'll have Sumeet Rai presenting, followed by Adrian Cioanca. Did I say that right, Adrian? Do you want?
It's good enough.
Okay. I wanted to check with you before. Okay. Fabulous. Sumeet's going to start with us. Sumeet's a intensive care specialist at Canberra Hospital and a PhD candidate in the School of Medicine and Psychology. Sumeet researches the healthcare burden of intensive care survivors. We're thrilled to hear Sumeet's talk today, titled "Psychological and Health-Related Quality of Life Outcomes in Ventilated and Non-Ventilated Intensive Care Survivors, a multi-center prospective observational cohort study." And Sumeet, I'll go ahead and just stop sharing my screen, and you can take over when you're ready.
Thank you very much, Nicole. Thank you for the kind words, and thank you for allowing me to present. I'm just going to try and share my screen. Right. Can you hear me all right? And can you see my screen?
Yes, we can. Thanks, Sumeet.
Okay, excellent. So I'm going to talk about this part of my PhD study on the long-term outcomes of intensive care survivors. And one of them is specifically looking at the psychological and health-related quality of life. And to give you, so firstly, I have no conflict of interest. To give you some background, and I'm sure most of you understand this in the audience, that the intensive care unit looks after some really sick people. They require a lot of support when their organs are failing. This could be in a number of ways. When your lungs are failing, you could be on the ventilator. In that time, you're supported by a number of things as well. And over the years, we found that survival has actually been improving across the board internationally, especially in Australia and New Zealand. And that's because we've got better at doing intensive care, because the community's got better at primary health care prevention strategies as well. But unfortunately, we found that over the years, the group of survivors has created almost like a survivorship burden, which they have a lot of ongoing issues. Some of these are described as post-intensive care syndrome. They're broken up into physical, mental, and cognitive issues, and these persist for a long period of time. There's some studies which have followed these patients, intensive care survivors, for up to 5 years, and found that they still have persistent issues. Now, some of you have probably not heard about post-intensive care syndrome, and some probably have. And you're gonna, I'm sure, have thoughts about, is this the same thing as long COVID that you've more commonly heard now in the last few years? To some extent, yes, there is definitely an overlap between the post-intensive care syndrome and long COVID syndrome. And I guess long COVID is more popular, but it is kind of like a type of post-intensive care syndrome anyway. This first, the term was coined only in 2014, but over the years, there have been a number of studies, and I'm going to focus on the mental health outcomes And a number of studies have longitudinal follow-up studies have looked at the survivors of intensive care who have post-traumatic stress, anxiety, depression, and a number of other cognitive issues over the years. In fact, a significant meta-analysis done on these as well. The post-traumatic stress varies in literature. I mean, it goes from anywhere to 4% to 60% in literature among survivors of intensive care. But by far, I think recent studies would probably quoted at around 1/5 of survivors having post-traumatic stress symptoms at longitudinal follow-up around one to 2 years. Whereas depression and anxiety is also not uncommon in intensive care survivors. And around 1 year, 1/3 of the patients will probably have depression or anxiety symptoms to be noted. Whereas, what about Australia? Recently, there have been, in the last decade or so, some studies that have come out, mostly from Victorian researchers, they've looked at the long-term outcomes of ventilated intensive care survivors, and have found, again, a varying literature, a varying incidence of PTSD, anxiety, depression, varying from anywhere PTSD around 8 to 16%, around 14 to 25% of depression, anxiety. However, I think intensive care is more and more admitting patients who do not require invasive ventilation. So if you look at a snapshot of the Australian, New Zealand intensive care practice, only 35% of our patients are invasively ventilated with a breathing tube in their throat. A lot of our patients come for therapies that do not require invasive ventilation. Some of them are managed with a non-invasive ventilator with a mask on their face, some with high-flow oxygen therapies alone to help with their breathing, and some just require invasive support for their blood pressure or circulatory or hemodynamic support, as we call. But mostly a lot of them come in for high acuity nursing care and monitoring needs as well. The literature, though, doesn't really describe this vast majority of patients with their long-term outcomes in great detail. And there's no significant longitudinal follow-up study which looks at this non-intubated survivors. So we wanted to have a look at part of the study is what is the outcome of the non-intubated intensive care survivors, and how does that compare to the intensive care survivors who are intubated, which we know a reasonable amount on. So when we designed this, it was supposed to be a multi-center prospective observational cohort study, and we followed these ICU survivors over a period of 12 months, and we a priori divided them into the intubated and the non-intubated group. The idea was to compare the severity of the psychological symptoms as the primary outcome, and then secondarily to look at the health-related quality of life between the groups. There were 4 tertiary units, the Canberra Hospital being the primary one, and the Royal Adelaide Hospital, and 2 hospitals from New South Wales, Nepean and John Hunter. And we started screening for intubated and non-intubated patients, and the screening period was between 2016 and 2018, with that last cohort requiring a 12-month follow-up period, which ended sometime before COVID. Those are the criteria for the inclusion criteria. So the intubated survivors had to have been ventilated at least for 24 hours, and that way we were kind of and stayed in intensive care for more than 72 hours. And that way we kind of excluded those patients who sometimes come to us after just a surgery where they haven't been able to be woken up or had delayed awakening and take a few 5 to 6 hours to get extubated. So we kind of excluded that less severity of illness. So these were sick people in the intubated group. And the non-intubated group, you had to have some kind of an organ support. So either an inotropic support or a vasopressor support to help with your blood pressure, and or some non-invasive ventilation to help with your breathing. There was some exclusion criteria. You had to be an adult. Background psychiatric history we did exclude, although some other studies have included patients with background psychiatric history as well. And because it was a longitudinal follow-up study, we excluded patients who were imminently likely to die, and those who couldn't speak any English, because most of the screening tests that we were using were validated only in English. And if they were unable to give us informed consent prior to hospital discharge, we couldn't include them. So what was the follow-up like? The follow-up essentially started at baseline. So with the psychological tools, we screened them first at a baseline to get us an idea about where they were at. We did this after they got discharged from the intensive care unit, but before they got discharged home. So up on the wards. With the health-related quality of life, it was only at follow-up. The follow-up periods, as I've explained, is 3 months and 12 months. For PTSD, we used a UK PTSS-14 screening tool. This is a well-validated tool recommended by the UK Intensive Care Society for Long-Term Follow-Up. This is validated against the kind of the gold standard post-traumatic distress scale, PDS. It's a 14-item scale. It goes from a score being 1 being never any problems to 7 being the worst problem. So a minimum score of 14 to the highest score of 98. And a clinical significant cutoff score is usually counted to be as around 45 in the literature. We use DAS-21 for depression, anxiety, and stress. And the DAS-21, it's a 21-item short scale, which is then converted to the 42-item DAS scale. It's got depression, anxiety, and stress. The scores going from 0 to 3, and there are severity markers for the depression, anxiety, stress based on your scores. The severity goes from up to, I think, 5 scales. For health-related quality of life, use EQ5D5L, again, a validated tool. It has 5 main scales, or mobility, self-care, usual activities, pain, discomfort, anxiety, depression, and each has 5 levels. So you can have 1 for no problems in a level. So you could score yourself as 1, 1, 1, 1. Which is you're completely fine, that you don't have any problems, to 5, 5, 5, which is the worst problems you could have. These scores are then converted to a utility scale, which is used according to the Australian population norms, or whatever local population norms you have. And then that kind of gives you an idea about what the scores in the population is and what the comparison is. And there also on the right side, you can see a visual analog scale, which is a self-rated scale that patients would mark depending on what their health is like. Right. We did publish the protocol and the statistical analysis plan previously in 2019. But what about the results now? We screened a number of patients, but we included only 172 patients. We had powered this study to look at the difference between the intubated and the non-intubated group. And our power calculations around 75 in each group, which accounted for a 20% attrition rate, which is common in long-term ICU follow-up studies. Unfortunately, what we didn't factor in is the amount of the mortality we would have in the non-intubated group. Which was much higher than in the intubated groups, because we had really sick people in the non-intubated group as well. So at planned follow-up, we had 133 patients, but after death and some decline of participation in the groups, we had 116 and 3 months. And then finally at 12-month follow-up, we had 94 patients, because there was a much higher mortality in the non-intubated group, and a number of the non-intubated group people finally declined to participate at that 12-month follow-up. Most of them had participated in the 3-month follow-up. And so that's our patient characteristics. As you can see, mostly slightly elderly population, a lot of them males, 10% indigenous population, but mostly a medical population. So, which means these weren't those elective surgical patients by far. A lot of them had respiratory failure in this, and a subcategory sepsis was around the 25% mark. As you can see, a number of them have had a lot of comorbidities, and some of them, in fact, had multiple comorbidities. And the non-intubated group, interestingly, had a lot of malignancy, a lot of patients with active malignancy in that. If you look then at the ICU specific data, the patients in the intubated group were more sicker. They had a lot more delirium. So 32% of them had some delirium in intensive care compared to 4% in the non-intubated group. And the Apache scores are severity of illness scores used in intensive care unit. They were much higher in the intubated group as well. Also, the hospital length of stay and ICU length of stay was also much higher in the intubated group. From discharge destination survivors, most survivors went home. A number of survivors in the intubated group needed to go to rehab before they could be sent home, but hardly anyone in the non-intubated group went to rehab. So what did the results show? From a PTSD screening point of view, the PTSS-14 scores were much higher at baseline. Now, I'm sure most of you would know that we don't really look at PTSD within a month of the traumatic event, but this just gives us an idea about the trend. So we weren't counting whether they had PTSD, we're just looking at the trend from the baseline, and the baseline scores are high, but then gradually come down over a period of 12 months. But as you can see, the intubated and the non-intubated group, there wasn't any difference between the two groups, and statistically there was no significance as well. But we're looking at around 10% population which has PTSD. Sorry, just 1 second. So it has PTSD in both groups and overall, especially. So this was graphically representing that trend, which shows that the scores were coming down over a period of 12 months. What about depression? Again, scores are higher at baseline, but they came down. But and overall, 20 to 25% of the population still had depression, persistent depression at 12 months. When you especially looked at the DAS scores, we used the moderate cutoff. So that's a score of 14 or higher. So we did not look at those who had no depression or mild depression. We just thought, compared to literature, moderate depression on the DAS kind of correlates with the HAD score that is used internationally, and that will give us a better idea. And there was still a reasonable amount of population with moderate depression at 12 months. Again, the scores were all coming down over a period of 12 months as well. Similar things with anxiety, very high at baseline, and then coming down over a period of 12 months, but at 12 months, no difference between the two groups, and anxiety around 19% in the survivors. With stress, again, moderate stress, reasonably high initially, but then around the 10% mark at 12 months. And again, no difference between the two groups, and scores continue to come down over the 12 months. What about health-related quality of life? Now, we looked at the 5 domains, and what you've seen here is that out of the 5 problems, the 5 levels that people have on EQ5D, we dichotomized it into no problems or some problems. And these were all patients who had some problems in those domains. So there was a widespread, problem in each domain, but the intubated group at 12 months almost twice as high was in the mobility, usual activities, or anxiety depression domain than the non-intubated group. But the visual analog scale or the self-rated scale, there was no real significant difference between the two groups, the intubated and the non-intubated groups. And how does this all compare to population norms? Well, if you remember, what I said is that PTSD in our study is around the 13%. The population norms, Australian Bureau of Statistics or the recent surveys done is around the 5% mark for PTSD. So definitely more than twice of population norms. Same with depression, we're actually 4 times higher. The population norms for depression around 5%. Anxiety, the population norms around 16%. Our study survivors at 12 months at around 19%. So there is a significant incidence of long-term effective symptoms. And as compared to ICU survivors, this is similar to international literature, but there's no difference in symptoms between either the groups. And over 50% of the patients had problems with their quality of life, but the intubated group had a higher and persistent problem with quality of life, especially in mobility, usual activities, and depression. So what are some of the strengths? So it's a diverse case mix of survivors. We use non-intubated survivors, though there's very little literature on this, and there's the only literature around that is in some cases of people doing studies on patients with non-invasive ventilation. We had an acceptable, I wouldn't say a low overall loss, we had an acceptable loss to complete follow-up, which is consistent with the ICU literature. And we had baseline measures, which very few studies have done, and that allows some kind of observation of trends as well. We had a number of limitations, though. We had screening tools. These aren't diagnostic tools. The diagnostic tests take far too long to diagnose PTSD, anxiety, depression in ICU survivors. And we did, malignancy was more common in the non-intubated group. And while I've not shown the risk factors, there wasn't any significant risk associated with that when you account for malignancy. Unfortunately, we didn't have any data on that trend in the health-related quality of life because we didn't collect baseline health-related quality of life because it's hard. Baseline health-related quality of life expects patients to think back and have potentially some recall bias because they have to think back about what their health-related quality of life was before they came into hospital. So we didn't specifically collect that, but there is literature on data which says that it's probably not a bad idea to do that to allow monitoring of trends. What are the implications? Well, there are significant psychological health-related quality of life. Unfortunately, this leaves a kind of a lasting legacy on some survivors. We need further long-term follow-up research, especially on this increasing group of population who are not really intubated in intensive care, and need to look at their risk factors as well. But to conclude, we do have significant long-term psychological distress in ICU survivors. The non-intubated survivors, though, who've previously been excluded from long-term follow-up studies are no different, and they actually should be included in those studies. But intubated survivors do have worse health-related quality of life in all domains. Thank you very much. I'll stop sharing now.
Thank you, Sumeet. That was really interesting. I've just popped a note in the chat box for any questions. If anybody has any, please, you can unmute yourself actually and ask the question. We have good audio in the room that we're in today. Otherwise, I'll also check if any questions in the audience at the moment.
Meet is Jenny Gordon. In Goulburn, I'm the manager of the South East New South Wales Regional Training Hub and clinical nurse by background. I found that really very interesting that the non-intubated and the intubated outcomes were very, very relatable and very similar. So thanks for the study.
No worries. Thank you for that comment. I do have a limitation on that, which I should have mentioned, that we were underpowered to study the difference because of the significant fallout from the non-intubated group, either due to mortality or due to decline to participation at 12 months. Sorry, I think Carly has her hand up as well, but thank you, Jenny. Thank you for those.
Thank you.
Hi, Carly.
Hi, Sumeet. Thank you. That was really interesting.
Don't ask a tough question. I'll personally.
No,
say
I'm not. I'm not going to ask a tough question. I'm going to ask, you know, for a long time, we've really thought about the drugs being a significant contributor to a lot of this longer term psychological stuff for patients. And we traditionally don't give our non-intubated patients quite as much, certainly sedation. And I guess I wondered about your thoughts around some of that, that theory, I guess.
Yeah. I think benzodiazepines are obviously very notorious drugs like midazolam, diazepam, which are used for sometimes sedating and managing patients in intensive care. They're associated with significant delirium. And in fact, there's some incidents that they may be associated with worse psychological outcomes. Fortunately, in NZ, we use much less benzodiazepines for sedation. But having said that, I think why I didn't present it, benzodiazepines as on a logistic regression weren't really associated with any significant long-term outcomes, especially in the ventilated group, which would have received more anyway. But
any medication? Any medications significantly associated or none?
For the intubated group, oh God, I can't remember now.
Sorry, it doesn't matter. We can talk about it another day. Beg your pardon.
I've pushed it back to a subsequent paper just because this was too much data for a paper. But then there was something, but I can't remember now.
Fine, you can tell me another day. Thank you. That was really interesting. Great work.
Thanks, Carly.
Thanks, Carly, and thanks, Jenny. Sumeet, I just had a quick question related to the baseline outcomes that you actually had for some of the depression scales that you used. Can you just remind me when that baseline was actually taken? Was it when they entered hospital, but prior to any surgery, or was it at a specific time point?
No, so by baseline, I mean, they're probably not technically baseline because they were after the intensive care. But before they went home. So while they'd recovered from the intensive care, usually in a period of 7 to 14 days, we would follow them up and would try and get a baseline idea. And mostly because we wanted to see a trend. So technically, yes, you're right. It's anomalous to call that as a baseline because it's not before their ICU admissions, but it's more baseline before we started the follow-up.
Sure. I was just interested because you can imagine that prior to them having any type of surgery, they would have maybe been a bit anxious or had some level of anxiety just kind of going into that, or maybe depression from whatever issues they might have been having. And then similarly, following that, it would make sense why they're a little bit inflated. So I was just interested in the reasoning behind that a bit. But
yeah, that's a right point. Most of our patients, though, so if you look at that 60% medically, they're unplanned. The elective surgery are the kind of planned admissions to ICU, but because we only, even if they were electively surgically admitted post-op, we only included those that actually required some organ support. So they were already sick, and we often wouldn't know which ones they would be to be able to take, I guess, pre-ICU admission, kind of the baseline screening. So we just did it after ICU, but just before they went home.
yeah, it's a bit difficult with that population. Amazing. I think we'll move on. Thank you so much, Sumeet. That was fabulous, and we very much appreciate it. And sorry you couldn't be here in person. I know you really wanted to be, but hopefully next time we can all meet you in person as well.
Thank you very much.
Okay. All right, we will move on to our second presenter, which is Adrian Cioanca. I definitely did not say that right earlier, but you were nice. Adrian Cioanca, thank you. Adrian's presentation is titled Harnessing the Power of siRNAs to Combat Retinal Inflammation and Degeneration. Adrian completed his undergraduate degree in Advanced Vision Science at UNSW and then pursued a PhD here at the Australian National University with Riccardo Natoli in retinal degeneration. Following the completion of his PhD, Adrian learned bioinformatics and computer programming and is currently a postdoc researcher in the Clear Vision Research Lab with Riccardo and team. Adrian leads a research team focused on transcription factor therapeutics for the treatment of retinal degeneration. If you could just give me 1 second, I'll get his slides up and ready to go. Okay, I'll hand it over to Adrian.
Thank you for the introduction. So human lifespan has been going up and up, so we all live longer, but as we push the boundary of human lifespan, we become more susceptible to age-related changes. And most of our biology is affected by aging, but in particular, our eyes are really not immune to changes that occur later in life. And in particular, one part of the eye, which is the retina, it's a neuronal tissue, it becomes highly susceptible to age-related changes. And then if this tissue becomes degenerated, then we are likely to lose our vision, and losing vision, anyone can imagine, is probably not very fun. Now, what we've seen over the last few years is that really the likelihood of losing your vision as you age increases the further we go along the human lifespan. And right now, approximately 200 million globally are affected by this one disease of the retina, which is called age-related macular degeneration. And in Australia alone, we have about 1.5 million people that over the age of 50 that are affected by this disease. But really, if you look at people that are above 80, then approximately 20% of people of that age are likely to be affected by this disease, and they're likely to lose vision to some extent. So you can imagine that as we become older and older and we live longer and longer, the likelihood of losing our vision only because of this disease becomes larger and larger. And up until the beginning of the year, we didn't have any drugs that were actually able of reducing the likelihood of losing vision in this as a result of developing age-related macular degeneration, or AMD. Fortunately, around the beginning of this year, there was one drug which was approved. It appears to slow down the disease, but there are many side effects that are associated with this drug. So really, we are still requiring new therapeutics to be able to tackle this disease. So stating statistics about vision loss often doesn't capture the difficulties that come with the loss of vision. And by looking at the utility of vision and assessing that, what we have identified is that for when people are suffering detrimental effects to their vision, they are willing to trade 1 in every 3 years of their remaining life to be able to have their vision restored to a previous level. So this sort of highlights the impact that vision loss has on quality of life. So statistically, vision loss appears as a big problem, but really for the individual, it is a very large difficulty that might face, and we really need treatment to be able to tackle such diseases. Now, AMD has 2 different phenotypes. I won't go into the clinical aspect of AMD in this talk, but the kind of AMD that we haven't been able to treat up until the beginning of this year, and now we have a rather limited treatment that accounts for approximately 90% of the cases. For the vast majority of AMD, we still don't have a good way to treat this disease. Now, that's not to say that we haven't tried. In fact, we tried many therapeutics to treat this disease. We just haven't been successful. And we can really lump all of these therapeutics together by saying that their rationale is trying to suppress inflammation. Because we know that inflammation in the eye is really not good to have, because it does actually cause severe damage to neuronal tissue, which doesn't regenerate. So if you have inflammation into the eye, you may be able to prevent some damage because of other reasons, but really once you actually damage neuronal cells, they tend to not regenerate in the eye. So these vast lists of treatments that you see here, most of them have actually not been able to show any protection against dry AMD. So there's something that's not quite right with this, because we know, for example, from animal model, that if you are able to suppress inflammation in an animal model, you can actually reduce the severity of vision loss. So why is it that treatments which are good at suppressing inflammation are not able to provide protection in the eye? And that's because it's believed that because inflammation is actually a very complex process which has many moving parts, if you want to treat an inflammatory phenotype in the eye, the molecule that you use needs to actually be able to target multiple simultaneous processes that occur. And what we've been trying so far are mostly drugs which do not target multiple facets of inflammation. In fact, they're quite selective to only some biological responses that occur within an inflammatory environment. Now, it still makes sense to treat inflammation, because we know that if we are able to suppress inflammation, well, we should be able to provide some protection to neuronal tissue against damage. And the retina has multiple cell types, and it's a complicated but very structured organ. But when it comes to inflammation, there are two key cell types which are driving the inflammatory response. And those are both glial cells, and glial cells are cells which actually help neurons to do their job. And here we can see the two main glial cells in the retina, which are called Mueller glia or Mueller cells, and microglia. So microglia are professional immune cells that really are capable of driving strong inflammatory responses. And Mueller cells can both facilitate an inflammatory response, but they are also involved in a lot of other processes in the retina that maintains the architecture of the retina, and then it ensures that neurons, which are the cells that are able to pick up light and then also send that to the brain, their function requires also on the function of Mueller cells. So in a way, if we want to find a therapeutic that targets inflammation, then we need to make sure that that therapeutic has an effect on the glial cells of the retina, but it doesn't really affect the neurons, because we want our neurons to be able to function in a way to do their very specialized functions. So inflammation is really occurs in the retina because it's trying to prevent some damage that might occur because of some injury, but in doing so, it actually causes a lot of like collateral damage, and then it's affecting neurons which cannot be regenerated. So moving on, if you want to understand inflammation in a model system, we have to be able to induce inflammation to be and then look into the processes that are driving that response. So our view to find new ways of targeting inflammation is to initially model that in an animal model, and then explore the biology of that animal model to identify the molecular aspects of inflammation. So what you're looking at here is one of the model systems that we have to be able to trigger an inflammatory response in the retina. So what we do is we use mice for our work, and then we expose mice to high intensity bright light. And the exposure of the eye and the retina to light induces degeneration, and then a part of that process is the increase in inflammation, and another process called gliosis, which is a change that occurs specifically to Mueller cells, and part of that change results in the formation of molecules that drive inflammation. So what you're looking at here is a healthy retina of the mouse, and then in red we see a particular marker which is elevated when there is increased stress in Mueller cells. So here we can see that there is very little expression of this red marker across the retina, but when we cause stress to the retina, this expression of the protein goes up, and that tells us that Mueller glia are now in a state where they can drive inflammation. On the other hand, microglia, this other type of immune cell, what they do in an environment where there is a lot of neuronal stress is they migrate to the area that is affected by light, and then they begin to actually phagocyte to also kill the cells in that area. So here we can see in microglia that they actually migrate towards the part of the retina where the photoreceptors are. So these are the cells which detect light. And then when microglia get to this particular layer, they actually begin to damage it. And then that's one of the reasons why we lose photoreceptors. And by losing them, we're not able to see. So in this model, we see the sort of the typical presentation of a pro-inflammatory state in the retina. So because we are able to induce it, then we can actually study it and understand why it happened, and then maybe find ways to prevent against this process happening. So even though inflammation is a complicated process, we can really get a very clear understanding of this process by understanding gene expression, because any change in our biology is underlined by change in gene expression. So that's exactly what we've done. We've taken our animal model, and then we looked at gene expression in healthy and in animals that have been exposed to this, to light, to induce damage. What we found is that approximately 5,000 genes were either going up or down in expression. So this large amount of change illustrates the complexity of the biology that underlies the degeneration process. But it's important to say that not all of these 5,000 genes regulate inflammation. There's a lot of things that are happening, but clearly somewhere within this complicated gene expression profile that must exist the mediators of retinal inflammation. And then what we've done next is we took these 5,000 genes, and then we know which molecules drive inflammation. And then we were able to identify that approximately 700 molecules that are changing in response to damage in mice are involved in some part of the inflammatory response that we see here. So now this gives us a potential list of molecules that we might want to target if we want to be able to suppress inflammation. The problem is that we do have to target a very large number of genes. And for context, previous therapeutics that we have actually tried to get working in people were targeting just one or two molecules that are involved in inflammation. So this really is a complicated process. So we need to find a way they can target multiple molecules that are driving the process. So there are not that many ways that you can really use to be able to target a large number of genes at the same time, but there is one possibility for doing so by using transcription factors. So transcription factors are molecules that are able to access the DNA of a cell, and then they control which genes are being expressed in the DNA. And this is a fundamental mechanism that exists in any cell, and then this is how a cell maintains its identity, and then it facilitates the function of that cell. So really for a cell to, say, drive an immune response, what needs to be happening is some transcription factor binds to its DNA, then it will result in the production of genes and then proteins, which are driving that process. So these kind of molecules really have a great potential for treating very complex biological mechanisms. Now, the problem is that if we want to use a transcription factor approach to target inflammation, we need to know which one of those transcription factors we want to actually target, because there are many of them that exist in biological systems. But just to illustrate the power that transcription factors actually have is, if you know which transcription factor you want to, if you know which one you want to induce a biological change, you can, if you find the right combination of transcription factors, you are very likely to succeed. For example, you can take a skin fibroblast or a stem cell, and you can convert that cell into virtually any other cell. You can go from a skin cell to a neuronal cell simply by knowing which transcription factors you switch on and off within that cell. In fact, in a sort of more recent work that was done in the eye, it's found that you can even reverse the age of neuronal tissue in the eye by switching on and off the right transcription factors. So in terms of potential, transcription factors really give us a way to target very complex biological system. There's a point where we can even change cell identity by knowing which one of these molecules to use. The problem is that when we are trying to select transcription factors, I don't know why this is so blurry, but when we're trying to find the right transcription factor to target, we need to find a way to know which one of these multiple transcription factors that exist in the retina are really driving inflammation. And through the use of computational models, what we have been able to do in our data is to take this gene profile and then predict which transcription factors are really driving its expression. And then we found that a particular transcription factor known as CDBPD is one of the key transcription factors that actually targets regions of the DNA which are involved in inflammation. So we think that by modulating this particular molecule, we may be able to actually suppress the inflammatory response that occurs in the retina. That was at least the theory. So when we looked a little bit more into more detail into the retina, we found that this particular transcription factor doesn't really work alone, but it has a number of other friends, and they coexist in a family of transcription factors. And when we looked at the data initially, we looked in healthy animals and animals that were affected severely by the degeneration phenotype. But instead, what we've done next is we knew that this transcription factor is involved in the late stages of the disease, but we wanted to know whether or not it is one of the early changes. Is it really driving that early inflammatory response, which then leads to damage later? And then, so instead of looking at in control animals and 5 days later, which is when we see severe damage, instead we looked in control animals at 3 hours, 6 hours, 24 hours, and all the way up to the 5 days time point where we see lots of damage. And what we found is that this particular transcription factor, which we now know that is highly important for regulating inflammation, actually changes in its expression from around 6 hours of damage. And it's important to know that at 6 hours, we see very, very few changes with regard to the inflammation present in the tissue. So we think this is one of the earliest changes that actually initiates the inflammatory phenotype. And then when we looked at multiple variants of the same transcription factor, we found that 3 of them are in fact starting to be expressed from 6 hours onwards. And then we believe this is one of the early changes that may be driving the inflammatory response. And therefore, if we can intervene early, we might be able to suppress inflammation. Now, as I said before, if you are trying to develop a therapeutic that uses transcription factors as its main molecule to mediate the biological response, you want to make sure that that transcription factor is really not found in other cell types, because by changing or modulating its expression, you can have changes that are not favorable. And then by using an approach, by using single cell sequencing, where we can actually explore gene expression across all retinal cell types, what we found is that this particular transcription factor, it is exclusively present in mural glia and microglia, which are the key cells that drive the inflammatory response. So it's an early change. It changes in the key cell types, therefore it might be a biotherapeutic target. Now, of course, everything that we found so far was using animal models, and animal models are helpful in understanding diseases, but they don't always model all the changes that occur in diseases. And to make sure that the same change is also present in the human retina, we looked at the RNA profile of human retinas from patients that have donated their eyes. What we found that indeed in people that have AMD, they have a high expression of the same molecules, the same transcription factors. So our animal models identified it, and then the human data confirms it. So we think this is a really important change in the actual disease too. Now, so far we identified an interesting molecule. It can potentially be used to reduce inflammation, and it can be potentially used to reduce the severity of AMD. So what we've done next is design an experiment where we thought, okay, so if we can inhibit this molecule in our model systems, can we protect against damage? And essentially that's what we found, that it can be done. So we used an siRNA, and this is a kind of molecule that can suppress the expression of a gene. So essentially what's happening here is we suppress the expression of this transcription factor, we expose our animals to damage, and then we look to see how well they can see how well their retina functions. And what we found is that, animals that have received the treatment were more capable of detecting light, so they have a better response to light. Then more importantly, when we look in the area where a lot of the damage happens in this model, there was more photoreceptors in that area, so the integrity of the tissue was preserved when this transcription factor was suppressed. And this was really good to see, because now we know that we can actually have a functional change in our model simply by modulating this transcription factor. So that was good to see. And then what we looked at next was, okay, so we see a protection, we see an improved physiological response, but is that because there is a reduction in inflammation? And one way that you can quantify inflammation is to look at how many immune cells you find in the area where damage occurs. And what we found is that by inhibiting this transcription factor, there was a decrease in the number of immune cells in the key area where damage happens. And we sort of speculated that is one of the reasons why there is a protection against degeneration. Now, the last thing that I wanted to mention is that, doing research in the lab and identifying key molecular players, it's only one part of the process that's required to be able to drive a therapeutic from lab into the patients. And really part of that process involves being backed by large companies that can actually facilitate the clinical trials and so on and so on. And what we've seen recently is that there is a huge interest in backing the companies that are developing pipelines which can identify molecules to target transcription factors. So I think right now we are in an environment where we can really take basic science and potentially get help from the biotech sector to drive the development of new therapeutics for complex diseases using transcription factors as one of the key molecules to target. And lastly, I'd like to thank the whole Clear Vision Research Lab, and because it's run so quickly, many people are not on the slide. So I will not go through all the names in the interest of time.
Thanks, Adrian. Any questions from the crowd or online?
So I mean,
Yeah, it should just. Questions from the crowd? Most of the Clear Vision group is here, so that's fine.
They've heard this not long.
Yeah,
So
that's fine.
I should apologize instead, really.
I have a question.
Yeah.
I'm very curious in, well, this might already be happening, so excuse my naivety about this area, but I'm very interested in at what point do bioinformatics and AI join so you can find out, or are you already doing this with your computation and modeling and stuff where you can really pinpoint specific molecules, or AI is directing you to that site?
I mean, AI is helpful with like understanding really large data sets.
Yeah.
I
feel like at this point we have a lot of genetic data sets out there, but maybe they're not in one space.
Yeah, I mean, a lot of it's focused on like saying like the DNA sequence.
Okay.
And what we're really trying to do is look more at the RNA expression, because the RNA is what drives biological changes.
Yeah.
And as technologies become more and more available, yeah, we can use them to be able to like really understand the biology in a lot of detail.
Yeah.
And it's also getting cheaper, so it is more feasible to do.
Yeah.
So we've done this on a small number of samples, but really we are trying to like apply this like very large patient cohorts and see what happens there.
Yeah,
Mm.
fair enough. I have another question.
Sure.
Nobody else does. So you mentioned they did develop a drug earlier this year,
Mhm.
but there's a lot of terrible side effects. And inquiring minds would like to know what are the terrible side effects?
So
And do you think there's gonna be newer drugs that are coming out a lot more?
we sort of think about AMD as being like binary into that stage where we've got like this dry phenotype that we couldn't treat, and we've got this other thing which is called wet phenotype, and the phenotype is marked by the block of blood vessels into the retinal that damages the tissue. And what they found is in this particular study when the drug was approved is that it slows down the dry AMD phenotype, so it doesn't advance as quickly, but it creates a kind of vision that makes you more susceptible to the other phenotype, the wet phenotype. And the issue, and bit of a trade-off, and then the issue is that the wet phenotype is actually far more aggressive. So while dry AMD develops over 20-30 years, and at some point you lose vision, with a wet phenotype, you're going to probably lose vision even within months.
Yeah.
And then, while there are some treatments for the wet phenotype, many patients are not responsive. Trying to treat one phenotype, you might make patients more susceptible to the other phenotype. And they also have to go to 2 rounds of injections into the eye, which still there's issues there too, because it's expensive
Yeah,
and so on. So that was one of the observations from the study, but that wasn't part of the original hypothesis in terms of approving the drug or not. And apparently that's why it was a clinical trial.
Yeah.
Primary outcome was met, therefore the drug was approved. The secondary outcomes were not part of the approval process. So that's something for the patients to be aware of if they think they want to. And it's not available here, yes, it might be.
is that in the US?
That was not in the US, and often it's sort of positioned, yeah.
Sure.
Yeah.
Okay. Fascinating.
Mhm.
Any other questions? Questions online? Just got some comments. Fantastic research. Brilliant. Thank you so much, Adrian. You made it.